Well, good evening, everyone, on this fabulous Game 2 of the World Series. So thank you. You all deserve a round of applause for making it here tonight. We do appreciate your commitment to the ocean and to the New England Aquarium Lecture Series, of course. I am Gina Cerrito. No, I'm not. I'm Gina Parenti. That's my maiden last name. I haven't said that in a while. This is our fifth lecture of our 2013 season. We're really excited that you all could be here in our old home, the Harborside Learning Lab. We are normally in the IMAX. There's an event in there tonight, so I'm really glad that you all found us. We have a great season. We hope that you all join us again. Our next lecture is actually on Saturday here in this room. It's our Ocean Stewardship Showcase, featuring some great folks from Boston Harbor Cruises, our Live Blue ambassadors, which are our teen volunteers, as well as some great people from the Boston Harbor Islands as well. And there's free lunch afterwards, so please join us. And that takes place again here Saturday at 11 o'clock. And for tonight's lecture, Joe Blasi is Manager in Visitor Education at the New England Aquarium. Having cultivated her love of the oceans along the coast of Maine as a child, Joe studied biology along the coast of Connecticut and received her master's degree from the University of Massachusetts, where she studied the impacts of an invasive crab species on intertidal habitats. Her interests in combining education and marine science then led her career here at the New England Aquarium. During her tenure, Joe has worked with teachers in the Teacher Resource Center, taught summer campers as part of the Harbor Discoveries Camp, helped lead a group of teenagers on an immersion diving program in the Bahamas, and now can mostly be found teaching aquarium visitors around the wonders of penguins, sharks, or New England habitats. Please help me welcome my colleague and very good friend, Joe Blasi.
Thank you everyone for joining us this evening. I know Game 2 is going off in about an hour, so we'll make this not quick but timely. I thought I would start off by talking a little bit about my trip to Antarctica with a little bit about Antarctica. I didn't really know that much about it other than sort of what you learn in grade school. It's usually that sort of white blob on the bottom of the map. It's one of the continents, kind of cold. It's actually a pretty cool satellite image. And here's what it looks like from space. And the part of Antarctica that I'm going to be mostly speaking about this evening is this section here. And it's called the Western Antarctic Peninsula. It's a piece of the continent that sticks out towards South America. And that's the part of Antarctica that I travel to. But some other details about Antarctica: it is the 5th largest continent. It's twice as big as Australia. It's also one of the windiest and driest places on the planet. Particularly in the dry valleys of the interior, there's very little precipitation that happens throughout the year. On the Western Antarctic Peninsula, there actually is a bit of precipitation throughout the year, so it's a little bit different in that respect. Ice covers about 98% of the continent, and that ice actually constitutes 70% of the world's freshwater. It's a huge amount of ice and freshwater that's in that system. It is pretty cold. Average temperatures at the South Pole, minus 56 degrees Fahrenheit. Again, the peninsula is a little bit warmer. And one thing that I didn't realize when I had started this is that not one country can claim Antarctica as its own. It's governed by a treaty that's signed by over 50 countries and allows for a peaceful research area in its waters and on the continent itself. Now, for the U.S. Antarctic program, there's 3 research stations with this program. The first one is McMurdo. It's the largest and probably the most well-known. This is the one that people get through New Zealand. It's over on the Ross Sea. The second is the station at the South Pole, which they work with a lot of other organizations in other countries. And then the smallest station is Palmer Station, which again is located here on the peninsula. Palmer Station only has about 45 people at it during the summer season. During the winter season, it goes down between 15 to 20 people. So it's significantly smaller than McMurdo, which at any time might have more than 1,000 people located at it. In addition to the 3 research stations, there's also 2 research vessels, the Nathaniel Palmer and the Ellen Gould. And throughout the summer season, which they are just now beginning to get to the start of, there could be close to 3,000 personnel working throughout the summer and throughout the continent. Now, I was part of a particular program through National Science Foundation called the Palmer Long-Term Ecological Research Project. LTERs, as I will probably refer to it throughout the evening, are areas that are set aside for long-term data collection. So in order to study and find out changes in an environment, particularly sort of ecological changes, you need a really nice long data set. You can't compare a couple of years. You sort of really need decades upon research. And this particular research area was established down in the peninsula around the late 1960s, early 1970s. Palmer Station was established in the late 60s, and research quickly started after that. And the original sort of questions that the LTER wanted to find out is, how is the ecology there sort of regulated and changed by seasonal ice? It's not completely ice covered all year round. There is some open water during the summer. And really, how does that seasonal ice change things? But as they sort of saw some changes happening, particularly with the peninsula warming up, the question then turned to, well, what is the sort of impacts of climate change happening in this area? Is it really changing because of climate change? The peninsula is one of the fastest warming areas of the planet. In the past 50 years, they've documented it warming up 6 degrees, which is 5 times faster than the global average. And in the Palmer Long-Term Ecological Research Area, 9 out of 10 glaciers are receding and retreating. So the warming in this area is really pretty severe. So instead of saying, is the change happening, the answer is yes. Now the question is, well, what are those changes going to do? And sort of what are the mechanisms behind that change? Is climate change impacting food webs? Is it impacting nutrient cycling? Is it impacting some of these deep water canyon hotspots, which we'll talk about in a minute? And these are the questions that the scientists there are trying to answer now. So what sort of changes are we talking about? One of them, they've seen a decrease in sea ice, both along the peninsula and along the Palmer Station itself. So if you look at the top map, this is from 1975 all the way into the 2012 area. The blue line represents the sea ice average for the Western Antarctic Peninsula. The black line is specifically the area around Anvers Island, where Palmer Station is located. And you can see sort of an overall trend of decrease in sea ice as you go. Things like currents are also changing. With those glaciers retreating and melting, there's a huge influx of fresh water into that marine system, which could potentially input and change mixing patterns of nutrients from deep water areas. They've seen changes in weather patterns, which they're worried can also cause different mixing of water layers. They've seen changes in krill and phytoplankton distribution. When those sort of building blocks of the food web, things like penguin distributions, can also change. And the graph on the bottom is also a graph that was pulled off of the LTER data site. This is the number of Adelie penguin adults and chicks located right near Palmer Station from 92 all the way up into 2010. And again, you can see both for the black, which are chicks, you see a decrease, and for the blue adults, you can also see a trend overall downward. So they know these changes are happening, and again, they're trying to figure out what's causing them. So here's the Western Antarctic Peninsula. We studied along the western edge of the peninsula. You'll able to see from north to south. That top aerial is where Palmer Station is located. And we traveled about 700 kilometers down the peninsula to Charcot Island, which is right here. We made a stop halfway at Adelaide Island, which is where there's a British research station. We were able to collaborate with colleagues there. So we sort of started up at the north, traveled 700 kilometers south, and made the return trip in all at about 32 days. Now, I had sort of mentioned these deep water canyons. Throughout the Western Peninsula, there are these deep water spots. If you look at the graph again, which was just a data site spot off of the ship's sensors, here's the water depth, and if you look very carefully, it goes from surface all the way down to 3,300 meters. It's over 9,000 feet. And there were some deep water canyons that were actually a little bit deeper than that in the study area. Now, these deep water canyons are really, really important areas. There's a process called upwelling that happens. Now, in New England, we have upwelling in Stellwagen Bank right off our coast. Cold, nutrient-rich water rises to the surface and helps sort of jumpstart the food web. Here, it's warm, nutrient-rich water. The water is so cold near the surface due to the atmospheric conditions that the warm water is the responsible one for bringing up the nutrients. So this warm, nutrient-rich water rises up to the surface, it comes in contact with sunlight, and that helps jumpstart things like phytoplankton blooms. Now, these areas are so important. They've sort of been thought of as biological hotspots. these areas where they see aggregations of krill, they see penguin colonies nearby, and again, because of the mixing and the weather patterns and the temperature change and the freshwater influx and temperatures moving, they're wondering that whether or not this mixing can continue or whether or not that's in jeopardy. So, data sets been collected for decades and decades. Now, how do you make sure that you study the same spots in the same way year after year? Well, the way that the LTER has designed it, it's set up on a grid system. So it travels from north to south. So north towards the southern part, here's Charcot Island that we talked about earlier. Palmer Station is here. Adelaide Island is right in the middle. Goes from north to south, and the stations also go from east to west. So the coastal areas are along the eastern side, and it moves offshore beyond the Deepwater Canyon areas. So for each of those lines, these are 600, 500, 400, and so on. We would study 3 sample sites along that grid. Then we would move down a line, study 3 sample areas move down a line, and so on and so forth. Now, in order to sort of get more in-depth study areas and to be able to make sure that scientists were able to conduct experiments in particular areas, we also did what we called a process study. We did one up in the north near Palmer Station. We did one in the mid-grid, and we did one again down near Charcot Island. And again, those, instead of traveling between those stations, we would stay in one spot for 3 or 4 days, allow people to do any sampling they need to do or experiments they needed to do while in those stations. Now, one thing that's important to remember is that the LTER crew, it's a very short window of time for these scientists to get the data they need, the samples they need, so it was a 24-hour work cycle. Now, some of the labs on the ship would have people work a 12-hour shift and then a 12-hour shift off. Some labs, you were just up whenever you needed to do the work. The latitudes and longitudes that were assigned to each of these sample spots, as soon as we hit that spot, that's when you sampled. So the first day of science we did, I think I worked 20 hours straight. I got a 4-hour nap, and then I had to do it all over again. When the science had to be done wherever it was needed to be done, again, because the season is so short. And the logistics to make the season happen are really, really complicated. So I was simply amazed at the amount of time and effort and sheer will that drove these scientists and the researchers to get all of this done. It was really amazing to watch. Now, as part of the LTER and that long data set, there are certain areas of study that happen consistently. There's sort of 5 main components, and they are organized and run by different laboratories that come together to work on the ship. There's a physical oceanography component that's run by the Mont Doherty Lab, a group out of Columbia University. There was a biochemical and microbiology component, and this cruise was done by Texas Tech as well as Woods Hole Oceanographic Institute. The Phytoplankton team, which I was fortunate to be a part of, was organized through Rutgers University, Virginia Institute of Marine Science, which is the graduate program for the College of William and Mary, were the ones that studied the zooplankton. And then Seabirds is the 5th component, run by the Polar Oceans Research Group, which is a nonprofit husband and wife pair that have been studying penguins and seabirds in this area for over 2 decades. Now, we were also fortunate to have sort of 2 guest components on the ship. So these are not components that are a normal part of the LTER, but the goal, in particular for the marine mammal, is to actually become part of that. Things like whales and other cetaceans, as well as seals, are obviously a huge piece of that food web. And the group from Duke University was really trying to make an effort and make a case that they needed to be included on the cruise every year. And I hope they do that because they did a lot of really, really amazing work. So here is our transport as well as research platform. McMurdo as well as the South Pole research stations have an airfield nearby, so they're able to fly supplies and crew in and out. Palmer Station, which you'll see in a minute, sits on the edge of a glacier and does not have an airstrip. So the only way to get supplies in and out, as well as crew and researchers in and out of Palmer Station, is by the Gould. It's 230 feet, reinforced hull, so we can get through the ice. It holds about 45 people, about 26 scientists as part of the LTER cruise, and this was home for 6 weeks. We left from Punta Arenas in Chile, which is its home port, and it usually took, depending on the weather, around 4.5 days to make it to Palmer Station. We did have to cross the Drake Passage. For any of you who sort of know your maritime lore, the Drake Passage is one of the most inhospitable places of the ocean. with waves that can top 30 or 40 feet. We were extremely lucky on both the trip there and back. We had very, very good weather. There were all sorts of people telling me horror stories of trips that they had gone on, but we were very fortunate that the seas were relatively calm, which is good. Now, while you're going there, you do have to travel through the Straits of Magellan, which is like another big geography thing from grade school I was all excited about. While you're there, there are some beautiful sights, lots of birds to take pictures of. We had some amazing birders on the boat, and we all got an amazing set of bird lessons. The top one is a turn taken in the Straits of Magellan, and as soon as you come out of the Straits and start making your way south, there's not much except gray ocean and gray sky and birds. So lots of pictures of birds on this trip. Now, during the transit, scientists are also setting up their laboratories, getting ready to do research, unpacking equipment, making sure their lab spaces are set. And then after 4.5 days, you land here. So this is a picture I took of Palmer Station. It's not much, but it is an amazing place to do research. You can obviously see the big glacier behind it. It's a set of buildings that has laboratory spaces, dormitory spaces, common areas for the staff. There's also a boathouse. They did a lot of research from boats. They were sort of reinforced zodiacs. And even though this is taken sort of in prime summertime, you can still tell there's a lot of ice around in the water. And depending on which way the tides are going or the winds are blowing, they could either get out to do research or they would have to wait a couple days. I just got a report that right now they're trying to get their season set up, and there's a lot of sea ice still down there, so they're having trouble sort of getting their season started because of that. And then you can see the Gould, Home Away from Home. So this is taken from Torgeson Island. This is the other sort of thing you can see on Torgeson Island. While we were unloading cargo for Palmer Station, we had a day layover essentially before we went out to do science. And there is a colony of Adelie penguins that live on the island, along with some very grumpy elephant seals who are molting. It was a gorgeous day to take pictures, really nice weather, beautiful blue sky. We were able to see a lot of penguins. So here's a gentoo pair down on the bottom. You can tell the gentoos with their little white earmuffs. But again, the majority of the penguins in this area are Adelie penguins, and you can see this mama with 2 chicks who are looking all sorts of fuzzy. By the time we came back to Palmer Station, these chicks had grown significantly and were just about ready to leave mom and dad on their own. Now, an interesting story, sometimes with change, it might hurt one organism but might benefit another. One thing that they're seeing in the Adelie populations right near Palmer Station is the Adelie penguins are having a little bit of a harder time. They like things a little bit colder, rely a little bit more on that sea ice. However, the gentoo penguins don't need that. The gentoo penguins live usually a little further north, and what they're seeing as the sea ice decreases, the gentoo penguins are able to come in and compete with the Adelies for breeding space. So they're seeing that a little bit higher gentoo populations than before, and the Adelie penguins seem to be moving a little bit further south. So speaking of penguin people, one of the nice things about being the outreach person on the ship is that you were able to sort of be involved and see lots of different research that was happening, and I was fortunate to be able to join some of the teams on some of their expeditions and see some of the research they were doing. We took a trip to Avian Island, where there's close to 75,000 Adelie penguins. We were able to drop a team of 2 off into this island with a bunch of penguins and seals, and their job was to count all of the penguins on the island. Good luck to them, but they did an amazing job. They were also looking at the chick abundance to see how the breeding season went. Here are some Adelie chicks that were obviously a little bit older than the ones we had seen right off Palmer Station. One of the 2 things that they did, in addition to counting abundance, was they did diet studies. Again, sort of the food web is being challenged. So here's a bunch of krill that they collected from a penguin stomach. The penguin nicely regurgitated for us. And they were able to see, what was interesting is that the krill that they would find from penguins returning to shore is that the krill was fairly intact, which means that they didn't have to travel out too far for that food. They were able to catch food and then be able to come back close to their nesting area. So again, those sort of penguin colonies near those deep water canyons are doing fairly well. Another thing that they're doing is the picture's a little hard to see, but they're satellite tagging this penguin. They want to know where these penguins are going out to forage. Are they using the canyons? Are they using the ridges right near the canyons? Where exactly are the birds going? And those satellite tags enable them to do that. Now, sort of the other charismatic megafauna that was studied on the boat were the marine mammals. So this is the team out of Duke University. So they were doing a couple of things. The first is that they were just looking at abundance and seeing what was there. The other thing they were studying was specifically the humpback whale population that's there. Very similar to what we do with whales up in New England. They were trying to photo ID them so they could join the humpback whale catalog, which is actually kept up in Maine. And what's also interesting about the humpback whales in Antarctica, at least in this section, is they travel in pairs. And they're not quite sure what the relationship is between those whales. So one of the things they're doing is DNA analysis. And again, very similar to what we do with our right whales here, is they can get a biopsy sample using a crossbow. So particularly in areas where we would be stopped for a while, we wouldn't have to be towing any nets, they would let the whalers off in their Zodiac. They would be able to go out and find some whales, and they were able to get a biopsy sample. And this is Zach, who's a student from Duke. He was an amazing shot with a crossbow. And this is actually one of his biopsy darts flying through the air. And he managed to get a fantastic sample. And you can actually see Dave Johnston from Duke University pulling that sample out. One of the reasons it made such a great biopsy sample is that if you look very carefully, there's sort of the black piece, and that's the skin sample from the whale. And they're able to do the DNA analysis of that. And there's also a little bit of blubber that's there as well, because they were starting to do lipid analysis to see sort of what was in that fat content, that blubber content, to see if they could figure out what the whales were eating. So we've sort of gone from the big down a little bit smaller. So this is some of the work that the Zooplankton crew did. This is what it's all about. This is Antarctic krill. They're one of the main food sources from everything from penguins to seals to whales. And they eat phytoplankton. You can see those nice big green bellies, all full of food. So sometimes the zooplankton research, all it entailed was dropping a net into the water and then being able to sort through everything, which was a huge task. A lot of my lab work meant spending some time in their lab, and they worked really hard to identify all these little different invertebrates. Now this picture here, this is Dr. Debbie Steinberg. She is she is a very important person, but she's the principal investigator for the zooplankton group. And this is her student, Miriam. And one thing that I like to remind people is that the ship was not strictly just PhD scientists. They also brought their research students with them. They brought lab techs with them. And a lot of the students were doing research of their own down there. They were participating with their lab and making sure that the lab got the data that the LTR needed. But Miriam was working on her graduate work with copepods. So she was down there supporting her lab as well as getting some of her own research done. And that's something really, really cool to think about. Now, phytoplankton is where the group that I was fortunate enough to be embedded in. Again, because the LTER was able to secure funding from National Science Foundation to do a more advanced outreach program, I was able to secure a spot on the boat. And Dr. Oscar Schofield and Grace Saba, who's also a PhD in the Coastal Ocean Observation Lab, they were able to donate one of their spaces on the cruise to me, and I'm very, very thankful that they did that. So in return for a bunk, I get to help them with their research, which was an amazing opportunity. Now, why study phytoplankton at all? Phytoplankton, little tiny plants, dinoflagellates, things that are photosynthetic in the ocean. And one of the things that's really important is that they're essentially building blocks. They're able to take nutrients as well as sunlight and turn that into organic energy that the rest of the food web is able to use. They're really, really important for that manner. So the lab is really interested in studying a number of different things. They wanted to know the concentration of phytoplankton throughout the study area. They really wanted to figure out what kinds of phytoplankton were around. Again, there's lots of different things that fall under that category. And they also wanted to assess sort of the overall health of the phytoplankton blooms that they saw. So we studied this both in the laboratory setting and also had some really cool technology to study it out in the field. And if you think about it, sort of phytoplankton, krill, and whales sort of form one of the shortest food webs that you could possibly imagine. So all really important. So this is the lab work. We worked mainly with a piece of equipment called a CTD. It's a conductivity, temperature, depth. It's this whole big contraption. The CTD is actually this piece of scientific equipment on the bottom. And then it's usually corresponding with a series of what they call Niskin bottles. And this rosette, I think, held 26 or 26 bottles. And what you're able to do with the CTD is send it down to the depth of wherever you're sitting at, and then use it as it's coming back towards the surface to collect water at very specific depths. Now this was important not only for my lab group, but also for the microbiologist group. There was somebody who was studying nutrients in the area. So they're able to grab all this water from different depths. And then what my lab was able to do was filter out the plankton from those samples. It was frozen in liquid nitrogen afterwards, sent back to Rutgers for study for that concentration classification based on the pigments that they found. And they were also starting to do some genomics work to see exactly what was there. We were also able to run water samples through what they call a fire instrument, and that essentially tested how efficiently the phytoplankton can turn sunlight into organic energy, so essentially how healthy the phytoplankton is. Now this is me just getting one little old water sample, but usually there were 6, 8 people all trying to get water all at the same time, and it's pretty funny to watch. So we have to get out of here. We have to go here. Depending on what lab shift you were in, sometimes CTDs collecting water samples were accompanied by song, by haiku poems. You get really creative at 3 in the morning when you're really tired and need to collect water samples. Why, hello. And we did about 70 of these casts, CTDs, over the course of the cruise. And then what we would do, another video, is that once we collected that water, we were able to bring it back to a lab and be able to filter it out for the plankton samples. It's a little chaotic. So when we're able to filter the water, you see it will get a closer picture of those filter systems. Oh, there we go. Perfect. So when we were able to go to the CTD, we were able to collect water in these opaque Nalgene bottles. The reason we wanted them to be opaque is we didn't want the phytoplankton to start reacting with the light of the room before we could start sampling it. What we do is put water samples through each filter individually, and we would know how much water we would be able to filter through until we saw some color on the filter, which is located right here. So we put water in the tube or in the funnel. It's able to go through the vacuum. The phytoplankton stays on the filter. The water leaves, and they were able to take the filters off, as you see below. Again, freeze them in liquid nitrogen, and then be able to send them back. So by knowing the amount of water that you filtered, you're able to figure out the concentration of plankton as we went. Now I mentioned we also did some field work with some pretty cool technology. Down at Rutgers, there's a series of Slocum's gliders that the Cool Lab works with, and Oscar and Grace both help with that project. Slocum's gliders are autonomous underwater vehicles. They look like little yellow drones. And what's really cool about these pieces of equipment is that they're buoyancy driven. So instead of having to worry about battery life, being able to worry about having a lower payload of scientific equipment in order for a power source, these the vehicles can stay out for 6 weeks at a time. And again, driven by buoyancy. Now they're controlled, which interestingly enough, through satellite by a crew back at Rutgers University. So these gliders were out for 6 weeks at a time. Every now would pop up to the surface, satellite back home to Rutgers. The techs at Rutgers would tell it where to go and sort of correct its coordinates, and then would go off again. And just like the sensors on the ship, they had things like a mini CTD, which is what's depicted in the picture. They're able to have optic sensors. They're even working on an acoustics and a pH sensor for later work. So really, really cool pieces of equipment. But why bother using them at all when you have all these people on this ship doing all of these CTDs? Well, remember, this is the picture of the original grid that I showed you, the sampling grid. For a CTD, we only did 3 stations along each grid. So for the 600 line, we did them sort of coastal, mid, and then offshore. So we only got 3 data points along this entire grid section. But the gliders that we had, they flew the entire line. So they were able to get data across that whole line where we were only able to get 3 data points. So what hopefully will happen is that the gliders are going to be able to take up a lot more of just getting the overall baseline data, and then the ships are really going to be able to be used as experimental platforms and to be able to do a lot more sort of in the field experiments rather than having to focus on catching all of that baseline data. Now, like any science project, there were some derailments along the way. The LMG Gould, the Gould was supposed to deploy a few gliders actually from the ship, but we were unable to do that. If you look at the top picture, this is the tail fin of a glider. You notice there's a couple of chunks missing. They believe that was because a leopard seal decided to go after the glider when it was in the water. They found it sort of stranded on a rocky outcropping. They were actually very lucky to find the glider. So we were unable to deploy as we had planned. However, they did deploy 3 gliders from Palmer Station. 2 of them studied the deep water canyon areas. One of them what they call mowed the lawn, actually went along that whole 600 line. And I'll show you what the data from that looks like in a second. One also studied Palmer Deep, which is a deep water canyon right off Palmer Station. And then there was a glider that ran Palmer Deep, ran along that deep canyon, but actually ran right along the top ridge. So it was actually a lot more shallow and looking a little bit more at the sort of phytoplankton blooms as they happened. Now I'd have to give credit where credit is due. The techs up at Rutgers did an amazing job. You have to remember these gliders were in the water for weeks and weeks at a time, and they had to time their flight with when the Gould would be in the area to pick them up. And they, again, have a very small battery. It's not like there's a huge window where the gliders can sit at the surface, you know, radioing back home, this is where I am. So the techs were able to fly the gliders towards the area where the LMG was headed. The gliders sort of surfaced, 2 of them surfaced, I think, within 10 minutes of each other, within 300 meters apart, which is amazing considering these machines were in complete opposite areas. And like I said, we were able to pick all 3 of them up successfully, which is, again, amazing flying by the techs at Rutgers. Another video. So this is what the glider recovery looked like. And again, the gliders were out in the ocean. We had people up on the bridge watching for them. Once they were spotted in the area, we knew they were at the surface. We hopped into the Zodiac and off we went. It's not easy when you're in a rocking Zodiac. We were also fortunate to have a film crew on board. What's nice is that the wings actually detach from the glider. Then they're able to load them into their carriers. We were able to upload all the data that they had collected, and then they were packed up and eventually shipped back home. Let's go back, here we go. There we go. So all sorts of data was collected. A lot of it was not sorted out while we were on ship. Again, the point of the research is to get all the data collected, and then you have the whole next year to look at it. So this is the type of data we would get from one CTD cast. And again, we have this sort of data for 70 of the casts that we did. So it's a little bit hard to see, but there's a lot of data on this one little slide. The red line is temperature. Goes up. Things like oxygen, the blue line is measured as well. That's all well and good, but my lab is really looking at this line. So this is the fluorescence line, and this was able to tell us where the phytoplankton was blooming. And the phytoplankton here is sort of blooming around 30 meters below the surface. So we were able to, using this data as the CTD went down to the bottom, we sort of knew, okay, well that fluorescent peak is around 30-35 meters below the surface. We're able to collect water around that area, and that was the water that we would then sample in the laboratory. Now, one thing that it definitely showed that there were lots of fluorescent peaks, many of them sort of where the temperature increased, but it wasn't too close to the surface. One thing that they're trying to figure out is what's more important for phytoplankton. Is it the sunlight? Is it the nutrients? Is it a combination? And they were actually doing some experiments on that as well. But sort of one thing that kept coming to light is that upwelling is really, really important, again, along those deep water canyons, and that's where we saw the most productivity. Now, speaking of data and why gliders are important, this is what the data looks like for a glider. So this is the 600 line, that grid I had shown you earlier. The stars indicate where we did the CTD casts. So we just have those 3 points of data. The glider was able to capture all of this, both for salinity and temperature and other things depending on what the glider payload was. It's really amazing. We did 70 CTD casts, so essentially 70 points of data. The glider was able to capture 4,000 points of data. So the gliders are becoming an increasingly important part of getting a lot of that basic data that the scientists need to understand the greater part of their experiments. So where did we sort of find? Again, some of the work is still being done, but things that we did see, we saw those subsurface blooms between 25 and 30 meters below the surface. That's where we consistently saw a lot of phytoplankton blooms. And that's sort of where the warm, nutrient-rich water hits just a little bit of sunlight. As soon as you got too close to the surface, that productivity dropped off. If you got too deep away from the sunlight, it dropped off as well. But 25 to 30 meters apparently was the happy spot. There was higher productivity as we traveled south from sort of the northern part of the peninsula down towards Charcot Island. The concentrations and sort of what those phytoplankton types are is still being figured out. I'm sure the crew's been working really hard down at Rutgers working on that for the past year. But one thing we did see was something called Phytoplankton Antarctica. It's an algae species that's usually formed or seen in sort of singular organisms, but they will come together and form big clumps. And the reason we were able to identify this particular algae species while we were down there is that when it forms these big clumps, it's actually large enough for you to see it, which is unusual for a lot of phytoplankton. We could see it swirling around in our filter columns, and you could also smell it. It had a sort of sulfur smell to it, so even when you had the CTD come up and you were collecting your water samples, you would open a Niskin bottle and you could automatically smell that Phytoplankton. So it was found in an area that was a little unusual. They don't know exactly why that's happening. I'm sure that's something that they'll look at again this year when they head down, whether or not it was there, whether or not it was sort of a fluke. But that was something a little bit unexpected. So sort of next steps for the LTER and the National Science Foundation's Antarctic program down there. Again, the labs are continuing to sort through their samples, looking at the data from the 2013 season. They're going to be able to take all of that biological and oceanographic information and sort of correlate that with all of the experiments that they did. You know, one thing that's really nice and sort of a little different about the LTER is that there's so many different scientific disciplines on one ship. They're all doing research at the same time, in the same place, which is very unusual. So if the phytoplankton group had a question about what types of krill were in the area, we walked right next door. If the whale guys were wondering sort of what the population of krill was in the area, they went downstairs and talked to the zooplankton guys. So it was really nice to sort of have this cross-discipline approach, which makes it a really, really interesting program. So they're also being able to talk to each other, even during the off-season, to see what's going on. And some of the folks on the ship have been working together for a long time, so they're all very good friends. Thankfully, the shutdown is over. The Antarctic season was actually in jeopardy. At one point, they were about to declare caretaker status, which meant the season was going to be cut. All non-essential personnel and equipment were going to be pulled out, and ships were going to be returning home. But fortunately, shutdown's over, and everyone is sort of packing up and getting ready to head south. The LTER trip probably won't go until we left right after Christmas, and sort of spent Christmas to the beginning of February down there. So that's usually the time the LTER goes. However, there are people that are down in Antarctica or headed down to Palmer Station soon, if they're not there already, ready to do research on behalf of a lot of different other projects. And again, the logistics and the planning and the grant work and finding funds to go down is sort of a never-ending process, and they plan years in advance for these types of trips. When we went at the beginning of 2013, Oscar and Grace were already talking about 2015, 2016. The outreach program is now thinking on a 6-year plan ahead, so they're thinking long, long beyond just the season they're gearing up for. Another thing that's sort of been happening, sort of not related directly to the LTER, but there has been an international push for marine protected area around Antarctica. There's been some pushback from certain countries, but they're trying to come up with a compromise. One area they're trying to protect a lot is the Ross Sea. Even though the Antarctica is organized underneath that treaty, there's still a lot of fishing and things that are allowed in those waters, and there's trying to be a push to have those areas be protected from those types of activities. Now, if you're interested in learning more, definitely check out the Palmer website. They're about to unveil a brand new one. It's going to be pretty cool. On this site, you're able to keep tabs on what's happening this season, read profiles on all the scientists and what their work and what their focus is. So definitely check out the website. And another thing that sort of was a little bit different on this cruise is that we had a group from the Rutgers Film School taking a lot of footage of the LTER project. And they were going to be able to bring that back to Rutgers and create a documentary out of it. And that's in the process. You should definitely watch the trailer. It's called Antarctica Beyond the Ice, and it should be pretty amazing when it comes out. And you might even see a familiar face in it. Maybe, I don't know. It depends how they cut it. Hopefully they'll give me a good one. Now, just in case you were worried that was all science, I want to make sure that you are aware we also had some fun on the boat. Again, we were the same group of people together for about 6 weeks, and we got to know each other for good and for bad. So we celebrated things like birthdays. We had some fun out on deck. This is Dr. Grace Saba, totally mucking it up for the camera. It's a little bit difficult to see, but there's a bunch of us banging pots and pans. This was New Year's, so we decided to give the chief mate some company at midnight. He wasn't too happy, but we decided to have company anywhere. And this was one of the most picturesque places I can think of for having a soccer game. It was gorgeous, I know. We did have a couple of penguins who ran through the field. We had to sort of take a bit of a break. As I mentioned, sort of halfway through down the peninsula, there is the Rothera base, which is the British base. And they actually have an airfield, so they host us every year. The scientists get together, the rest of the crew have a little bit of fun. So they have an annual soccer game. Last year we won, which was huge, because the Brits usually beat the OMG crew pretty badly. So redemption was in the works. We only lost 1-0, but the Brits were so impressed with our soccer skills, they actually accused us of practicing, which is funny because we're on a ship. There's nowhere to practice, but it was a good time. And of course, there were tons of camera equipment on the ship. Pretty much if you had a free time between eating, researching, napping, trying to catch up on sleep, you were out trying to take a picture of something. There were lots of days where it was just gray sky and gray ocean, but then we also had some amazing sights. The icebergs, when the sun hit it, was just a completely stunning color of blue I've never seen in my life. This little fellow decided that our Zodiac was the perfect lookout spot while we were trying to pick up the field crew from Avian Island. He stayed there for a while, decided to hop in the water, and then another one took his place. It's kind of hard when you're trying to leave, when you have a penguin on board. One of the stops, we made a quick trip while there was a group doing some research to an abandoned Chilean research station, which is not so abandoned as there's a group of elephant seals decided to haul out there. These two males decided that they wanted to fight for about 45 seconds and went back for a nap. And then a Gentoo penguin giving his beauty shot. And sunsets, of course, were a fantastic time for people to gather up near the bridge and take some beautiful sunset pictures. It was light a good part of the day, being down in summertime. There was just a couple days where the sun dipped just below the surface and then popped back up, particularly as we went further south. And then we had some amazing shots as we came back north as well. So, and because I'm sure somebody out there is wondering, because one of the most common questions I get is, how's the weather? You think Antarctic and you think cold. Yes, this is me in a sweatshirt standing by penguins. I sent this picture home and people were very confused as to where I was. And they were like, but you're in a sweatshirt, it's too warm, I don't understand. So again, it's summertime in Antarctica. The peninsula is a little bit warmer. On this day, it was a gorgeous 45 or so degrees. On this day, when this picture was taken, when we were at Palmer Station, that was the really bad cold snap that Boston had. On the day this picture was taken, it was close to 30 degrees warmer where I was, and where all of you were. So we were fortunate. We had very, very good weather. We didn't have too many days of rough seas. The temperatures overall were pretty doable. You just layered, and you got to take some amazing shots with some amazing animals. One last beautiful sunset shot I will leave you with. Thank you very much for listening, and I'm more than happy to take any questions.
I'll let you read the question.
Yes.
Could you say a little bit more about what your role was as the outreach person?
Yeah, absolutely. So like I said, the LTER has an outreach component. And like I said, I was fortunate enough to be a part of that. They had received some funding from NSF to sort of put an educator out to sea. So the outreach coordinator contacted the New England Aquarium Teacher Resource Center and was wondering if they had a teacher or any ideas on how to recruit teachers. It's really difficult for teachers to take 6 weeks off at the beginning of a brand new semester. So I had a colleague who said, you know, this kind of sounds like something that might be up your alley. I'm like, okay, and I applied and went through the process and was fortunate enough to be picked. So part of what I did again was sort of help the Phytoplankton lab out, because they were able to graciously donate the space. I was able to donate my time to them. But my other responsibilities were for outreach. I wrote a series of blogs from the ship, both for the New England Aquarium blog as well as the LTER blog. So definitely check those out, they're pretty cool. A lot more pictures than I showed here tonight. I took 5,000 pictures, by the way. I couldn't show them all tonight. And then in addition to writing those blogs, able to come back and share my experiences in forums like this, be able to bring back pictures and film that the outreach coordinators are going to be able to use to develop programs, and be able to continue to share the science of the LTER with people back in the States. Any other questions?
More questions?
Yes.
What are the differences in the ice from year to year really apparent to the researchers who are coming back over and over again on the Gould?
I mean, the ice conditions, for as far as like logistics, you mean? Yeah, I mean, it can make a huge difference. You know, they never really know what the sea ice is going to do from year to year. Sometimes it comes in a little bit earlier, a little bit later. Sometimes it's really, you know, big pack ice, or sometimes it's just sort of chunky ice. So it all depends. You know, one of the big things is that if the ice is too socked in, they just simply can't get to their research sites. Or if you are in the middle of the winter and there's a huge ice pack right off Palmer Station, you want to make sure that the Gould can get to you, to make sure that you drop off supplies every now and again. So a lot of times, particularly when we were going to Charcot Island, Charcot Island is a site that they don't visit very much because they haven't been able to get there because of the ice. We were just fortunate enough that everything sort of worked in our favor, that the ice cleared out, that we can get to that study site. But sometimes, like right now, they're dealing with a lot of ice. They want to try to get to Torgeson Island. One of the cool things they're going to do is install a penguin cam in the Adelie colony, so you can actually watch your Adelies from home. But they're having a problem simply because the ice won't let them leave. Even though the zodiacs are reinforced, cutting through that ice can be pretty hazardous, and there are certain conditions that if the ice meets certain conditions, they're not allowed to actually leave station. So depending on what you're trying to do, where you're trying to go, and what ship you're in, the ice can prove to be a huge problem. I hope that answered your question. Okay. Yeah.
Why is Sharko Island called Sharko Island?
I have no idea why Sharko Island is called Sharko Island. That's an excellent idea. I have no idea. I should probably do some homework tonight, huh? Good question. You officially stumped me. Nicely done. I don't get stumped often. Good job. I had to guess it's probably named after somebody, but I don't know. Anybody know why Sharko Island is named Sharko Island? Anybody have a smartphone? Any other questions? Yes?
You mentioned the upwellings with Zooplankton Bay. Are you tying in any of the phytoplankton research to up here, or is it just too many different groups studying the different areas?
Yeah, I mean, there's definitely, people that are studying, Stellwagen Bank is a well-surveyed area. It's a marine protected area, one of the national marine sanctuaries, so it's well studied. One thing that, obviously they're studying the peninsula itself and sort of the ecology there, but, one of the things that, might in some cases be applicable is all of the changes that are happening due to increasing temperatures and the, decrease of upwelling and things like that potentially could be applicable to things like Stellwagen Bank or the Pacific Northwest, where all of those sort of same cycles and systems happen, but just haven't been impacted as greatly due to the temperature increase as down on the peninsula. So they're not necessarily making direct comparisons to other systems, but the research that they do down there could have big implications to other places where similar systems happen. Yes?
We were in the Ross Sea area.
Brilliant.
And they were talking about penguins
Mm-hmm.
and the sea ice moving out.
Yes.
I gather under the ice is a lot of the krill.
Yes.
And so they were trying to get a sense of what that was doing to the penguin population
Yeah.
in that area. I just wondered if that was part of the concern when you were there.
Yeah, I mean, well, they're definitely looking at, we're obviously there beyond the sea ice has already sort of come and gone, which is why, we're able to get into those spaces. That distribution of sea ice and the krill relying on that is definitely something that they're worried that, not only are the Adelie penguins being able to have that additional increase of the Gentoo penguin population coming in, but having to change and move their feeding sources. There's some historical record that the Adelie penguins were eating other things, including small ice fish that were there, but those populations have left, so they've switched over to krill. So if the krill disappears, then are they able to adapt and eat something else? Again, one thing that they've seen is that like the avian island population of Adelie penguins seems to be doing okay. The population near Palmer Station is not, it's decreasing. So are the Adelies just moving south? Are those populations doing better? There is a very small population on Charcot, I think of about 60 or 70 birds, and they're keeping an eye, as close of an eye on that population to see will that population continue to grow in the future. So like I said, the LTR is nice because everybody sort of works in concert together, so they're able to talk to the zooplankton group and say, well, what did you find for krill numbers? Is it low? That's interesting. We've seen this for the penguin population that year. So again, some of the data that was collected, I don't know the results of because they did that, over the past several months being back home. But it's definitely something sort of historically that they've seen and are continuing to monitor.
Could I ask one more?
Sure.
You mentioned the phytoplankton, I think, at around 100 feet.
Mm-hmm.
And that's in open water. Is that different than the krill that's under the ice?
Oh, the different species?
Yes.
Yeah, like Superba versus what they call crystal krill. I'm not really sure. I don't know my krill species.
I get the impression that a lot of the feed was under the ice as opposed to the open ocean.
Yeah, some of it. I mean, they're still feeding during even the summer. Like I said, there's I think 3 or 4 different types of krill species in that area. So it all depends on the season and timing of things. The Rossi is obviously on the complete opposite side. The Rossi is the ones where, that have like the big emperor penguins and those sorts of stuff a little closer towards the interior. But yeah, I'm not quite sure about that specifically. You might know more than I do.
Not likely.
Any other questions?
We found out what Sharko Island is.
Sharko Island is
Two smart
named.
people operating smartphones over here.
It's discovered in January 2010. No, 1910. By the French Antarctic Expedition under Jean-Baptiste Charcot, into which it was named. So there you go.
I think that's
Jean-Baptiste.
a perfect way to end.
Exactly.
If you have any other questions, Joe's going to hang around for a bit. We have coffee and
Absolutely.
cookies in the East India room, just on the other side of the hallway. So thank you again for coming, and we hope to see you again soon.
Thank you very much, and go Sox! It's almost 8 o'clock!